we're going to be continuing where we left off from transcription to discuss the processing and decay of RNAs in the cell, as well as reverse transcription and other sort of unusual kinds of polymerases. So I actually wanted to start by just making sure that everyone was familiar with some terminology that I have been using and will be continuing to use. And this is to say that helicase is an enzyme that breaks base pairs. It separates two strands. Those can be two strands of a DNA double helix, or they can be, say, two strands of an RNA that's folded on itself, or they could be, strands of an RNA DNA duplex. But it breaks base pairs apart and does not break phosphodiester bonds in the backbone. By contrast, nuclease is an enzyme that breaks phosphodiester bonds in the backbone. And there are a lot of different helicases and nucleases involved in all of the processes involving DNA replication, transcription, splicing, and translation. And most of these have a lot of substrate specificity. And there are a lot of different kinds of substrate specificity they can have. Some of them are specific for, say, DNA or for RNA. And I guess one particular kind of specificity to talk about are exonucleases versus endonucleases. So I mentioned that nucleases break the phosphodiester bond in the backbone. An exonuclease does that essentially only right at the end of the molecule. Removes NMPs or DNMPs from the ends. Usually it's specific for one end, and it will remove them just from the 3' end or just from the 5' end. And often it's very specific for whether its substrate is single-stranded or double-stranded and which strand it removes. Endonucleases, by contrast, cleave the backbone in the middle. These also have specificity. And they can be sort of specific for, say, one nucleotide sequence or for one particular structure. So an example of an endonuclease that we discussed is the AP endonuclease involved in base excision repair that cleaves specifically at A basic sites. So just wanted to make sure we were all on the same page about those terms for enzymes. So if we're looking at bacterial RNAs, the ends of those RNAs, as they exist as mRNAs in the cell, are exactly the same as they come off the polymerase. And one note, of course, is so RNA polymerase, uniquely, is something that DNA polymerase cannot do. RNA polymerase can start a new strand. And when it starts a new strand, the first nucleotide triphosphate incorporated is not the subject of nucleophilic attack. And in fact, the 5' triphosphate is left on the end of that very first nucleotide. And in fact, that's the state of the 5' end of an RNA in bacterial cells for essentially the entire life of that RNA. And the 5' triphosphate is left on. And likewise, at the end, if you remember, termination essentially involves pulling the growing RNA off of the DNA template. There's no sort of special or additional chemistry that happens there. And as a result, the 3' OH that's present at the end of a growing RNA chain is simply what's left at the end of a bacterial RNA. And some of the other non-mRNA transcripts in bacteria are processed differently, and we'll mention that a little bit. But these are left essentially as is until they're degraded. And so bacterial mRNAs are typically very short-lived. They can sometimes live just 2 or 3 minutes. Their degradation happens through a 2-step process. The first is an endo-ribonuclease cleavage, so an RNase that cuts somewhere in the middle of the transcript, and usually this is an RNase called RNase E, and it tends not to cut at a region of the transcript that's being translated. And after you have that first round of endo-cleavage, there are exoribonucleases that chew up both ends. And all of these actually exist in a complex called the degradosome that contains this endoRNA, some exoRNases, as well as something that has an RNA helicase activity that can pull apart some RNA structure, so places where the RNA is formed a hairpin. It can separate those into linear molecules that are easier for the exonucleases to chew up. And so, of course, there's no nucleus, and as we'll talk about, that means that transcription and translation can happen simultaneously. It also means actually that transcription and degradation can happen simultaneously and on the same RNA that translation is happening. So all of these things can go on at the same time, and typically, the RNA is very short-lived and might not finish transcribing before it starts to be degraded. By contrast, eukaryotic mRNAs are processed extensively. And of course, eukaryotes are characterized by the fact they have a nucleus where all this RNA processing can happen before the RNA is exported and potentially translated in the cytoplasm. So if we look at the DNA of this ovalbumin gene, which is simply a gene that was studied very early in our understanding of splicing, you can see that the primary transcript is much longer than the final RNA, mature RNA that's translated. Over 75% of that primary transcript is spliced out into, in this case, 7 introns. The 5' end is processed. The 3' end is cleaved and also processed as well, and the coding sequence of this RNA is assembled from a bunch of discontinuous pieces in the genome by splicing. That means that when you sequence a eukaryotic genome, you don't necessarily find the protein coding sequences of all of its genes in order. And in fact, one of the big challenges in assembling genomes is actually figuring out which pieces get strung together to assemble the final coding sequence. So the first modification that happens to your eukaryotic mRNA is capping. So this is the 5' end of the RNA, and this is the first base that's actually incorporated by the polymerase. And what we find is there's actually a modified guanosine residue connected backwards to the very 5' end. So you can see the 5' phosphate on this is linked to this triphosphate group here is actually connected to the 5' end of the first base. So there's a 5' linkage, no free 5' phosphate, in fact, no free 5' end at all. This is exposing a 3' end, and of course this is the extending to the 3' end of the molecule. And people speculate that this can serve as a defense against viral RNAs or other parasitic RNAs that aren't going to have this distinctive structure. It's certainly true that the cap plays an important role in the stability of eukaryotic mRNAs as well as their efficient translation. So the chemical process of capping is sort of diagrammed here. So the primary transcript, the thing that the polymerase first synthesizes, has the same kind of triphosphate 5' end that we see on a bacterial mRNA. Very shortly after it's produced, though, one of the phosphates is hydrolyzed off of the end, and then this beta phosphate performs a nucleophilic attack onto a GTP here. And so this results in the guanine and one of its phosphates, its alpha phosphate, being connected to the beta and alpha phosphate of the first NMP in the transcript. And after this guanine has been attached, it's methylated with acetylmethionine as a methyl donor on the 7 nitrogen here. So in this cap, this part comes from the GTP. The methyl is added later from SAM. This comes from the first nucleotide of the transcript. There can be additional methylation that happens on the 2' OH positions of the first 1 to 2 bases of the mRNA as well. So all of this process happens co-transcriptionally. That the cap synthesizing enzyme is recruited to the C-terminal domain of RNA polymerase based on the phosphorylation status of RNA polymerase. As we discussed last time, this C-terminal domain is a repetitive sequence with a number of serines that can be phosphorylated, and which ones of those serines are phosphorylated changes over the course of transcription. And there's a specific phosphorylation state that's favorable for recruiting the cap synthesizing enzyme near the beginning of transcription. And this capping occurs, the cap synthesizing complex dissociates. There's actually something called a cap binding complex that then specifically recognizes this very unusual 7-methyl G connected backwards to the very beginning of the RNA and binds to it. We call this nuclear cap binding complex. There's a different one when the RNA moves into the cytoplasm. It's exchanged for a cytoplasmic cap binding complex. So one important consequence of the fact that capping occurs co-transcriptionally and on RNA polymerase II is that Pol1 and Pol3 transcripts are not capped. So Pol1 transcripts are the major rRNAs, the three largest rRNAs, and so those are actually all processed out of a single transcript in a totally different way. Pol3 transcripts are a variety of short and non-coding RNAs that are also subject to extensive cleavage and processing that are not capped and not polyadenylated. So after capping, splicing is the next form of processing. This is a diagram from the book. I just want to make the point that actually the way they drew it, it looks like splicing happens on the completed primary transcript. That's not true. Most splicing, there's a large amount of splicing, happens co-transcriptionally, and it's actually can be important that it does happen co-transcriptionally. So the process of splicing is removing these intron sequences, which can be substantially larger than the exons, and connecting together the 3' end of one exon to the 5' end of the next exon. And so when we think about it chemically, some parts of this are easy. It's an isoenergetic process, so you're just trading phosphodiester bonds back and forth. But there's no nucleophile here in the RNA to serve as a point of attack. Both this, the very end of the upstream exon, the very beginning of the downstream exon are already involved in phosphodiester bonds. There's also this question about what would be the exact chemical state of the intron. And before we talk about the sort of eukaryotic pre-mRNA splicing, I wanted to discuss what are probably sort of the ancestors of the eukaryotic pre-mRNA splicing, which are self-splicing introns. So these are very interesting RNA sequences. The first one that was characterized is called group 1, group 1 self-splicing intron. It was first identified as an RNA sequence that was present in the rRNA of tetrahymena. And so this sequence folds up into a very specific three-dimensional structure that's shown here. And the patterns, you can see that this three-dimensional structure is full of these helices, and the pattern of base pairing in all of these helices is illustrated in this diagram here. And then all these contacts that really assemble this into a catalytically active structure that can function as an isolated RNA with no protein components to catalyze its own splicing. The basic reaction is shown here, where this red sequence here is folded up into this very specific and characteristic shape of a group 1 intron and catalyzes the splicing of the 5' to the 3' exons of the tetrahymena rRNA and removes itself. And to look in a little more detail at that, this is a process with 2 steps. So I mentioned that this question of what is serving as a nucleophile in splicing. And the first step nucleophile for a group 1 self-splicing intron is the 3' OH of a guanine nucleotide. And so that 3' OH is held, actually, it's sort of drawn better here, that it's held in a specific position by the self-splicing intron to favor an attack of this 3' OH onto the phosphodiester backbone. So this cleaves the backbone, so leaving an intermediate that's actually in 2 separate RNA molecules. It breaks the backbone here. There's the intron with this G actually attached to the beginning of it, and now we have a free 3' OH on the 5' side of this gap. And in the second step, that free 3' OH can now perform a nucleophilic attack here to actually carry out the splicing reaction, which is the ligation of the very 3' end of this upstream exon to the very 5' end of this downstream exon. And the product has attached this extra G that served as the nucleophile in our reaction, has attached that to the very 5' end of the intron product. And the exact chemistry to carry this out is the same kind of chemistry that we've seen from RNA polymerases and from many other kinds of enzymes, that it's a nucleophilic attack by this OH onto the phosphate that then releases this and leaves a free 3' OH which then again performs the same kind of nucleophilic attack onto the phosphodiester bond at the beginning of the downstream exon. So these group 1 self-splicing introns were the first class of self-splicing introns that was characterized and discovered, and these are present in a few genes in many different organisms, including bacteria, and in fact in bacteriophage. There are also group 2 self-splicing introns, which in a lot of ways are similar to group 1 self-splicing introns. So the key difference is the first step nucleophile here is not sort of an extra guanine nucleotide that's sort of floating free. The first step nucleophile is actually the 2' OH of an adenine inside the intron. So this needs no other components, no other nucleotides, except for the ones that are actually present in this group 2 intron. And so this 2' OH attacks the backbone here in order to cleave this phosphodiester bond and leave a free 3' OH on the upstream exon, just like the first step of group 1 self-splicing introns, except the use of a different nucleophile. And so this actually creates a 2', 5' phosphodiester bond and leaves this adenine nucleotide here in a sort of an unusual structure where, of course, it's 3' and, of course, continues on to this exon, but its 2' end is also connected back here, 5' end here. And then when this reaction proceeds, we get a spliced RNA, and the spliced RNA looks exactly the same as the ordinary phosphodiester backbone, but we produce this lariat product that has an A. It's called a lariat because it has a big loop and a tiny tail at the end, sort of like a lasso. And the junction point of this adenine, is called the branch point because it's where the chain actually branches in 3 different directions instead of being a linear chain like 1. So again, this is an adenine with 3 phosphodiester bonds, 1 connected to the 2' OH. And chemically speaking, there's no really strong reason why the 2' OH is not just as good as the 3' OH. For making this connection, it certainly is unusual in RNAs, but it's sort of from a purely chemical perspective, it's just fine. So these self-splicing introns catalyze their own excision out of the transcripts that they come from. And actually I have a question about thinking about these self-splicing introns as catalysts. Go ahead and started that so these self-splicing introns are catalytic but are not true enzymes. And the question is which thing is true of as an explanation of why these things are not like actual enzymes. And the answer is that the self-splicing intron is not a true enzyme because it carries out this reaction only once on itself. Whereas an actual enzyme can carry out multiple rounds of catalysis of the same reaction. One protein molecule can carry this out multiple times. So splicing is an isoenergetic reaction, but in fact, enzymes catalyze reactions only in a favorable direction. The catalysis doesn't change the energetics of the reaction. Sometimes enzymes can couple reactions to something, couple a disfavorable reaction, something like ATP or GTP hydrolysis that is favorable, but it certainly isn't necessary. And some enzymes do catalyze things that are favorable reactions where simply the kinetics would never allow that reaction to happen without an enzyme to help them along. Actually, many enzymes have intermediates where they're covalently bonded to the enzyme. So this difference that each molecule catalyzes only a single reaction itself is the reason we talked about these as catalytic RNAs and not as true enzymes. So the eukaryotic mRNA splicing is mechanistically exactly like group 2 cell splicing. So we have these two, group 1 and group 2. The main difference we see between these is that in group 2, the first step nucleophile is a 2' OH of a piece of the intron itself, as opposed to a prosthetic guanine nucleotide. And the exact same thing happens in the spliceosomal reaction that is actually catalyzed by pretty complicated machinery in the cell. And one consequence of this is that the introns, as they are released from the RNA that are being spliced, are lariat products with this 2' linkage back to the 5' end of the intron. So splicing, as I mentioned, a substantial amount of splicing occurs co-translationally. And again, components of the spliceosome can assemble on the C-terminal domain of RNA polymerase to interact with the growing transcript. So the spliceosome is actually composed of 5 separate complexes. Each of these complexes is called an SNRNP, which people often pronounce SNRP. And so they're U1, 2, 4, 5, and 6. There is no U3. And each one of these complexes is sort of nucleated around an snRNA, so a small nuclear RNA, that's bound by a collection of proteins. About 50 proteins in total that come together to make up these 5 different SNRP units. And these sort of assemble in a specific order in order to catalyze splicing. So, one key question is, where is a eukaryotic mRNA spliced? So the exact position that's spliced determines the exact position of the protein coding sequence, and this is an extremely important decision in gene expression. If you don't identify the correct splice sites, you won't produce a proper protein coding transcript. And in the case of eukaryotic pre-mRNA splicing, one of the major determinants is base pairing between 2 of these snRNAs and the sequences that are right at the ends of the 2 introns. So the U1 snRNA base pairs right with the 5' splice site itself. There are 2 nucleotides that pair actually inside the exon. The most important 2 that are completely invariant are a G and a U base pair in the first 2 positions of the intron. And then the U2 snRNA base pairs with the branch point with this A located here and actually flipped out of the base pairing region. The significance of the fact that the 5' splice site and the branch point are recognized first and most importantly, and the 3' splice site is not sort of directly recognized by this base pairing, is the first step of chemistry actually does not involve the 3' splice site. The first step of chemistry is the attack of this A onto this nucleotide, onto this phosphodiester bond right there. So, first of all, it's almost certainly why this base is flipped out to make it more accessible. It's not involved in the base pairing double helix, as well as why these are the things that are recognized. So the 3' splice site, there are some nucleotides that are present at essentially all splice sites. There's an A and a G right at the end of the intron, again with some preference for a G and a U at the beginning of the next exon. So there's a little bit of constraint on the sequence of the exon, but most of the sequence information to specify the 5' and 3' splice sites in the branch point that's located right near the 3' splice site is present in the intron, and so the coding sequence can essentially contain whatever actually protein coding information it needs to contain. So the recognition of a splice site by the spliceosome proceeds first by the assembly of this U1 snRNA, which is going to base pair with the 5' splice site, and the U2 snRNA onto the branch point that's quite near the 3' splice site. And so this process does involve ATP hydrolysis. The ATP is being hydrolyzed by proteins that are involved in the process, and these are things like RNA helicases that are actually changing the RNA conformation of these snRNAs to favor the base pairing with their targets. Once these have been recognized, the other snRNPs are recruited. U5 comes in, and U4 and U6 come in together in a complex. And so this is all assembled. And then the next step is actually the removal of U1 and U4. And so again, this is an ATP-dependent process as well, and actually generates an active spliceosome. So the cells put some energy into the process at the beginning, and then the rest of it proceeds in a sort of a downhill, sort of directional fashion, where this active spliceosome catalyzes, first of all, the branch point attack, and secondly, the attack of this upstream OH onto the downstream backbone, and the intron is released in complex with some of these spliceosomal RNAs as a lariat. And the idea, the reason that it's interesting to look at some of these self-splicing introns, especially group 2 self-splicing introns, is that the process of catalysis by an RNA, in those cases, probably the ancestral state of this. You can see that these snRNAs are left base paired to the actual intron that's coming out, much like the catalytic parts of the group 2 self-splicing intron are left, of course, attached to the part that's actually coming out. I mentioned that splice site recognition is extremely important for assembling the correct coding sequence of an RNA. And there are actually cases where the cell can pick different splice sites on the same primary transcript in order to assemble different protein coding sequences. So this is the same primary transcripts that are drawn out in yellow here. Each of these green boxes are introns, and these little lines like this are how we indicate the splicing structure of one of these transcripts. And I'm actually showing two different versions here. The difference between them is that in the light green case, the one sort of diagrammed up top, we use this U1 will base pair with a 5' splice site here, whereas in the dark green case, U1 will not base pair here, and instead base pair with this one that's sort of further downstream. And so this choice leads to cutting out different pieces of the RNA, and actually leads to two different transcripts that could encode two different proteins. And this process can be, in some sense, sort of stochastic or random, but it can also be strongly influenced by the presence of RNA-binding proteins that might bind to one of these splice sites and keep the spliceosome from recognizing it, or might bind the RNA near one of these splice sites and favor the spliceosome recognizing it. So there's a lot of regulation at the level of alternative splicing to control which RNAs are produced. And there are many different ways this can happen. It's sort of diagrammed here based on the choice of different 5' and 3' splice sites to use to bring together the RNA. There are cases you can simply include or exclude an entire exon corresponding to an entire chunk of protein coding sequence that might be included or excluded from the protein. You can also use longer or shorter forms of the same exon. In some cases, you can actually entirely retain an intron. An RNA that's produced in that way usually no longer actually codes for a protein. So this can be used to essentially transcribe an RNA but process it in an unproductive way to make something that's essentially junk. It certainly doesn't encode a useful protein for the cell. And then there are cases where you can actually have sort of mutually exclusive alternative exons. So you can pick one of n from a set of exons. And there's a particularly famous case of this in a Drosophila protein called DSCAN, where the combinatorial choice of, say 1 of 50 from a few different exons actually leads to over 38,000 different splicing isoforms being produced from a single locus. So this can be extremely complex, and it's one important layer of regulation in eukaryotic gene expression. And then at the end of the transcript, so the 3' end of a eukaryotic mRNA is not the actually end of the thing that's polymerized by RNA polymerase. What happens is a protein called the cleavage and polyadenylation specificity factor recognizes a specific RNA sequence in the RNA that's being transcribed while transcription is still going on, and promotes the endonucleolytic cleavage of the RNA transcript. So the transcript comes off, and this then triggers indirectly termination of polymerase II well downstream. So there's a little bit of extra RNA that's after the cleavage point that Pol II synthesizes, but is then degraded. At the same time, right when cleavage happens, so the cleavage leaves a free 3' OH, and that is subject to polyadenylation. So there's an enzyme, poly-A polymerase, that catalyzes the untemplated addition of more adenosine nucleotides to the 3' end of the transcript. It works by exactly the same mechanism as RNA polymerase, so it doesn't have a template. It always takes an ATP and adds it onto the end. And this can be from 20 to 200 plus A's added to the end of every transcript. And immediately after those A's are added, they're going to be bound by a protein called poly-A binding protein that always binds to the 3' ends of an RNA. So there are, in addition to alternative processing that happens sort of in the middle of an RNA, there are ways that choices of alternative promoters and alternative poly-A sites can change the exact RNA that's produced. So here's a case where you can get a longer transcript that's made by an upstream promoter that includes the dark green exons as well as this common exon. And here, a different promoter that would start a different primary transcript here would include these and then a shared final exon as well. So you can begin a transcript in 2 different spots but actually have shared sequences downstream, or you can have shared sequences upstream that end in 2 different spots based on cleavage and polyadenylation. And in fact, there can be interaction between cleavage and splicing because both processes are happening co-transcriptionally. So a processed eukaryotic mRNA has all of these exons strung together, a poly-A tail that is added in an untemplated fashion. This is not encoded in the genome, but it's present on the RNA, and it's bound by poly-A binding protein and a cap binding complex binding to the 7-methylguanosine cap at the 5' end. And proper recognition of the transcript in the nucleus with a cap binding complex and poly-A binding protein is important for that transcript even to get out of the nucleus. So without these factors present, the transcript won't get out of the nucleus, and these also play an important role in the stability of the RNA once it does get out of the nucleus and into the cytoplasm. So eukaryotic mRNA decay happens mostly from the ends of the transcript. So there are 2 basic processes. First of all, there is the removal of the sort of the eukaryotic specific modification from one end or the other, followed by an exonucleolytic degradation of the RNA. So one thing that can happen is deadenylation, that 3' poly-A tail is chewed off, And once that tail is removed and there's no more any poly-A binding protein at the end of it, because there's no longer a poly-A stretch for it to bind, there's a complex called the exosome. It's a 3' to 5' exonuclease that can attack the 3' end of that transcript and chew it up from the 3' direction to the 5' direction. An alternate process that can happen is that the 5' end of the transcript can be decapped. That this triphosphate bond here, this very unusual linkage, can be removed, leaving just a single 5' phosphate. When that happens, there's a different nuclease, XRN1, that chews up the whole RNA from the 5' to 3' direction. So both of these can happen under different conditions. And there are also less frequent cases where either randomly or as a result of a specific enzyme, an RNA can be broken in the middle. And when that happens, the 5' end is immediately a substrate for degradation by the exosome, and the 3' end is immediately a substrate for degradation by XRN1. So after that cleavage in the middle, the RNA will be, both fragments of RNA will be very rapidly chewed up from that site in the middle, based on the fact that it does not have a 5' cap and a 3' end that are these sort of the markers for a proper mRNA in the cytoplasm. So RNA polymerase II makes mRNAs as well as some other non-coding RNAs in the cytoplasm. And one particularly important class of non-coding RNAs is made by polymerase II or microRNAs. And so we'll talk about these more when we discuss regulation. But microRNAs are short, about 20, 22 nucleotide RNAs that actually exist in an RNA-protein complex in the cytoplasm, base pair with mRNAs, and reduce their expression in a couple of different ways. And so the complex of a microRNA is this tiny little RNA with the protein factors that are responsible for silencing RNA expression is called the RNA-induced silencing complex, and this protein called arginine is probably the most important factor in here. So these microRNAs are not simply produced in the cell as a tiny 20 nucleotide transcript by RNA polymerase. They're actually produced as a large primary transcript. The primary microRNA, or pri-mRNA, is transcribed by Pol2 and actually is capped and polyadenylated in the same way that an mRNA would be. But then instead of undergoing splicing, it undergoes a different kind of processing. Each of the things shown in yellow is the sequence that's in the end going to become a microRNA. Each of those is found in that hairpin, where the RNA transcript folds back and base pairs with itself in a specific way that is recognized by this complex. This is called actually the microprocessor complex because it processes microRNAs, and the drosha is an important nuclease that is going to cleave those RNAs, the microRNAs out of there. So drosha binds these hairpins, it recognizes them, and it cleaves them into a precursor microRNA. So a single primary transcript can have several microRNA containing hairpins, and drosha will cut each of them out separately into a pre-microRNA. These things are about 70 bases long. They fold back on themselves, so they have about 30 to 35 bases on each side of this stem. There are two extra nucleotides to the 3' end. There's a 5' phosphate and a 3' OH. All of these features are very important for this thing to be recognized as a pre-microRNA as opposed to just some piece of junk in the cell. The pre-microRNA, which remember this is all still happening in the nucleus, at that point it's exported into the cytoplasm, and a different enzyme called dicer cuts the other side of the hairpin. So remember, drosha cut at the base. Dicer is going to cut off this loop. It's also going to leave a 5' phosphate and a 3' OH on a short RNA duplex that's shown here being stripped apart by an RNA helicase before it's actually assembled into this RISC complex. So all of this is sort of a very specific process in which the cell recognizes an RNA structural element in a primary call-to transcript and cleaves it at several places along a very sort of well-defined maturation pathway to finally produce this very specific microRNA. So we have another question that sort of comes from thinking about microRNA processing. So I mentioned that pre-microRNAs, as well as microRNAs, have a 5' phosphate and a 3' OH left on them. And this is actually just a true-false question, but the RNA cleavage, like happens in microRNA processing, always has to leave the phosphate on the 5' end and the free hydroxyl on the 3' end. Please go ahead. And here, just sort of for your reference, I've drawn up some RNA to think about how this might be cleaved.
It's a 50-50 shot.
Very strong.
I don't know. I think it's false. I don't know though. But I don't know. I straight up did. Amy, what is it? I don't know. Is it false just because? I don't know why though. I feel like it's false just because like always is a very strong word.
The answer is false. RNA cleavage very often leaves phosphate on the front end. It's actually just a drawing of RNA, of spontaneous RNA self-cleavage catalyzed by a base that we talked about near the beginning of this section, where here the 2' OH of the ribose is actually serving as a nucleophile. And I guess one point to make, sort of bringing this back to splicing, is that this 2' OH acting as a nucleophile is the same as a 2' OH acting as a nucleophile in splicing. So this is a good nucleophile. And then in the end, this leaves a 5' OH, and the phosphate is left and linked actually both to the 2' or 3', and it can end up on either in the end. And actually, this is the usual product of RNA cleavage by self-cleavage or by many RNases in the cell. And in fact, the 5' phosphate and the 3' OH that are present in a microRNA are actually different than the products of spontaneous RNA cleavage, and actually that's an important sort of marker used by the cell to tell a microRNA that was processed by the specific pathway, leaving a 5' phosphate and a 3' OH, from the products of spontaneous RNA hydrolysis, which leave a 3' phosphate and a 5' OH. So next I want to talk about some of these two processes that are sort of like asterisks on the central dogma, which are reverse transcription and RNA replication. So from a chemical perspective, these are actually totally boring. These are exactly like the synthesis of DNA with a DNA template, or the synthesis of RNA with a DNA template, except the template is an RNA in these cases and not a DNA. So in reverse transcription, it looks just like a DNA polymerase, except it reads the sequence off of RNA. In RNA replication, it looks just like an RNA polymerase, except it reads the sequence off of an RNA. Of course, the real thing that cannot happen is after translation of an RNA into a protein, that's where information is lost. But there's no sort of fundamental or theoretical reason why reverse transcription or RNA replication are particularly challenging processes. So reverse transcription arises mostly in the case of retroviruses, which are viruses that have RNA genomes and convert that RNA genome into DNA. RNA replication also can happen in RNA viruses with an RNA genome that directly copy their genome into another RNA molecule. It also can happen in this RNA silencing process that can be mediated by microRNAs. So this RNA silencing does not involve RNA replication in vertebrates, but in many invertebrates and in plants, there's a process by which an RNA can be copied into another RNA in order to essentially amplify the production of these short silencing RNAs. So this is a process that does happen in viruses, actually happens in many organisms. It happens not to occur in humans. And sort of the biology of reverse transcriptase, as I said, is that it allows a retrovirus with an RNA genome upon infection to make a copy of its genome in DNA. And it can then catalyze the integration of that viral DNA into the host genome. And so as a result, it permanently modifies the genome of the cell that it infects. And the integrated genome can be transcribed by the cell's own RNA polymerase. To make a transcript, it does two things. First of all, it serves as the template for producing viral proteins, but secondly, it actually serves as the viral genome itself. So the process of genome replication is actually done by RNA polymerase in the case of the retrovirus, which ultimately packages a new particle, the new RNA genome. So these reverse transcriptases were discovered first in retroviruses, and show a couple here. This Rouse sarcoma virus, it's a virus that causes cancer in chickens based on the way that the viral genome integrates into the host cell and changes gene expression. They also occur in HIV, the virus that causes AIDS, which is similar to retrovirus. And the interesting thing is there are also a class of retrotransposons. So these are essentially selfish genes, and they contain a reverse transcriptase. So the reverse transcriptase genes are called POL, or polymerase, as well as this GAG multifunctional protein. The retrotransposon doesn't include this N protein that encodes for the virus envelope. So a retrotransposon will transcribe itself, will be transcribed by the host, reverse transcribe itself, and then potentially reintegrate into the same genome, but it either has lost or never had the additional factors needed to actually produce a virion that could bud off of that cell and go to infect another. There is a sort of a specific prominent reverse transcriptase that's encoded by a normal cellular protein, that's telomerase. And we mentioned this before, and it solves a problem that comes up in lagging strand replication. And so telomerase is it's a ribonuclear protein particle, includes the telomerase reverse transcriptase as well as the telomerase RNA, is sort of embedded in this complex and serves as a template for the synthesis of repetitive DNA, this TTGDDG DNA shown here. And so it'll synthesize this 6-base repeating unit, and then telomerase will translocate down, prime, synthesize, translocate, prime, synthesize, those iteratively reused. And in fact, like all DNA polymerases, RT depends on extending a primer and can't actually start a new DNA chain. And so it's extending this end, walking down, extending the end again, and sort of iteratively reused. And this solves the end replication problem because now the linear end of a eukaryotic chromosome has, on the lagging strand here, an RNA primer that's degraded, but that RNA primer doesn't lose any irreplaceable genetic information. The cell can go in, use telomerase to resynthesize more of these useless TTGDGG repeats. And ultimately, actually, the structure in the cell was bound by some proteins that protect it. The cell normally recognizes any sort of break of the double helix, like to be present here, is a serious problem that needs to be repaired. And so these telomere-binding proteins recognize the ends, protect them, and ensure the cell doesn't try to repair them with the process that would use your double-stranded breaks by homologous recombination. And sort of putting together a few of the different things we've been talking about, I wanted to mention just at the end the presence of a reverse transcriptase in the self-splicing group 2 intron that essentially makes it into a retrotransposon. So the idea is the group 2 intron, of course, splices itself out of the gene that it's present in. And so it does two things. First of all, that spliced intron catalyzes its own removal, and secondly, inside that sequence is encoded a reverse transcriptase that can make a copy of that RNA back to the DNA. And in fact, it also contains an endonuclease, a DNA endonuclease, that's going to find some gene that does not currently have an intron, cleave the DNA, put the intron in, and then reverse transcribe a strand of DNA off of that inserted RNA. And so this whole process results in this new gene, gene Y here, having this element inserted. Now, of course, this is not going to disrupt the function of that gene, because when it's transcribed, this self-splicing intron cleanly splices itself out, leaving behind an intact RNA for gene B. And in fact, this is a speculation of how the prevalence of introns in cellular genes could have arisen, is that these introns are selfish elements that can copy themselves and insert themselves into new genes without disrupting the ordinary function of that gene. So that's what I wanted to talk about today, and